Welcome everyone to this seminar. Before starting our seminar, I would like to advertise a couple of initiatives. So the first one is for the trainees who are also members of our society. They have now the possibility to be featured on the society website. This is a very great possibility to improve your visibility inside our community, to know other early career scientists. So if other Cancer Cachexia Society trainee members would like to be featured on the website, please write to us, send in your biosketch. We encourage all the trainees to become members and part of our community, considering that the fee for the students is very low, accessible, and competitive. Second, all the trainee and the early career scientists can participate to our trainee talks. This is a very good occasion to share your data, to expand your network, to present yourself, if you are looking for a new position in the cachexia field, for example. It is a 15-minute talk plus 5 minutes of questions. So we will have 2 appointments, one in April and the second one in September. So how to apply? You can nominate yourself or nominate other trainees. The deadline is by the end of February. We will put in the chat the link that you received also by email. So regarding our upcoming seminars, so in March we will have Dana Brin from Pfizer. And the seminar by Dr. Morin has been rescheduled to May 15. So our speaker today is Sarah Lockie. So welcome, Sarah. And thank you for accepting our invitation. I know that it's very early in the morning for you in Melbourne, so we really appreciate you being here with us today.
Thank you, Roberta.
Welcome. Thank you very much. So Dr. Sarah Lockie is a neuroscientist and metabolic physiologist working in brain regulation of appetite and body weight control. Sarah completed her PhD in 2012, working at the University of Cincinnati and Monash University, investigating the physiological mechanism behind several classes of anti-obesity drugs. During her postdoctoral years, she worked with Professor Andrews at Monash University on the neurobiology of hunger with a focus on the orexigenic hormone ghrelin and its target within the brain, in particular the AGRP neurons in the hypothalamus. She is currently a group leader within the metabolism, diabetes, and obesity theme in the Monash Biomedicine Discovery Institute in Melbourne. And her current research uses genetic neuroscience techniques and mouse models to understand the failure of brain regulation of body weight and whole body pathophysiology in cancer cachexia. So her talk today is entitled Interrogating the Ghrelin-AgRP System in the KPC mouse model. So please, you can share your screen.
I think it should be this one.
Okay, perfect.
That looks all right.
Okay, another important information for the audience, you can type your question in the Q&A or in the chat, or you can also raise your hands. Please, Sarah, the stage is yours, and thank you again for accepting our invitation.
Thank you so much for the invitation, Roberta, and for the whole committee, from the whole committee. It's quite early here, it's 5 AM, so hopefully I will be firing on all cylinders, but if I'm not, you'll have to forgive me. So as Roberta mentioned, hang on, let me make sure I can. There we go. Good. Next
Yeah.
slide. Always helpful. As Roberta mentioned, I've spent my postdoctoral years really looking at the neurobiology of hunger and looking at how organisms adapt to, just let me, I've got an extra. There we go. Thank you. How organisms adapt when they are losing weight due to reduced food availability. And really, I guess the thing that I'd like to highlight about that is that during starvation or calorie restriction or fasting, we have a situation where there's really this coordinated, adaptive response that firstly protects the animal while it's in a state of negative energy balance, and then allows the animal to regain that weight once food becomes available again. And so it was from this background of kind of understanding how this works in normal physiological situations that I really became interested in why this is so dysregulated in cancer cachexia. Because on the face of it, if you look at sort of two animals or two people who are suffering these two conditions, they sort of look at a growth level relatively similar, but the specifics of the condition are sort of wildly different. And so this was really the difference between these two situations was what I was interested in investigating when I kind of started my own group and moving into my own research space. So as I said, cachexia poses an interesting paradox. Just to give you a little bit of background on how the brain regulates energy balance, we know that all of the systems that regulate energy balance are primed towards weight gain. So there are multiple overlapping homeostatic pathways that promote weight gain over weight loss, and these exist from millennia ago, where starvation was a much bigger problem for us as a species than overeating. However, now we live in a world where we've got this food environment, we can get Uber Eats at any time we like, and these pathways are predisposing us to eat more, both these homeostatic pathways and also hedonic pathways that drive us to eat because eating is enjoyable and because there are foods with sort of addictive properties of high fat, high sugar. And so our brains have been so effective at allowing us to put on weight that really we're facing an obesity epidemic, and it's not surprising to me that we are. It's almost the natural consequence of the situation that we've set ourselves up for with the brains we have in the food environment that we have. Oops, sorry. So the question then, sorry, early. So it's not surprising to me that we have this problem of obesity. So in the normal situation where we have calorie restriction or chronic undernutrition due to reduced food availability, we have a situation where circulating the hormonal profile of the organism changes, so that this is able to signal to the brain that we're going through, or a mouse is going through, a period of reduced energy availability. And this includes elevated circulating ghrelin, reduced leptin, insulin, and glucose. And so this hormonal milieu signals to this particular population of neurons in the arcuate nucleus of the hypothalamus, the MPY-AGRP neurons. And these are schematically represented here. They sit within this area of the brain that's really close to the blood-brain barrier, so they're able to act as sort of primary metabolic sensory neurons, and they regulate a range of physiological and behavioural outcomes to maintain energy homeostasis. And these include things like promoting feeding, motivated behaviour to find food, increasing locomotor activity, which is in the absence of food, that is, which is interpreted as food-seeking behaviour, and other things like improving memory and stress resilience that are broadly helpful to an organism that might be facing these issues with food availability. And importantly, these neurons contain the growth hormone secretagogue receptor, which is the ghrelin receptor, as well as leptin receptors, and are able to respond to this signal of reduced energy availability and transmit the signal to downstream neurons. And one of the primary mechanisms that they do this by is release of AGRP peptide, which is an antagonist at the melanocortin-4 receptor. And blockade of this receptor stops binding of this neuropeptide alpha MSH, which is released from this opposing population of neurons, the POMC neurons. These neurons are orexigenic, so they reduce appetite. So one of the main ways that AGRP neurons promote hunger is to block the binding of something that inhibits appetite, therefore increasing appetite. They additionally contain the neuropeptide MPY and GABA, both of which operate to promote feeding through their own receptor and downstream systems. So they're really very potent drivers of appetite. And we have this system that is conserved and that is that works really well. So what is going on in this Ghrelin-AgRP system that means that pathological weight loss is not sensed and corrected in cachexia? Why are these mice failing to eat, essentially? So to investigate this, I'm using the KPC model, which you're probably quite familiar with. So it's a model of pancreatic ductal adenocarcinoma. And these are the specifics of how we do it. I know there's quite a few variants on this model out there, but this is the cell line that came from Elizabeth Jaffe, and I've been working with Erica Sloan here at Melbourne at Monash Institute of Pharmaceutical Sciences to really have a look at, to sort of validate our model and make a decision about whether or not we used an IP injection or an orthotopic delivery of these cells. So for the IP injection, we've just suspended them in PBS and injected IP under light anesthesia. And orthotopic was laparoscopic surgery to implant the cells with Matrigel into the pancreas. So we really see when we image these using bioluminescence that the cells have a sort of trophism for the pancreas, and even with just injection of free-floating cells, we do get formation of these primary tumours in the pancreas that are really indistinguishable from the orthotopic implantation. So we've stuck with this IP injection model mainly for a couple of reasons. One, because it's a lot easier, obviously, to just do the IP injection, but also to reduce the surgical burden on the animals. And for me, setting the animals up for success, I suppose, in these models is important, because if you add a surgical burden at the outset, you're setting up an inflammatory situation in these mice, and then you're overlaying the cachexia phenotype on top of that. And given this is such an aggressive model anyway, we've really felt that the lower impact approach is working better for us. So just some validation data here. When we do the IP injection, they last about 16 days in our hands, but we've really started what's the word I want? Standardizing our collection at 14 days. It's because the last few days, the model becomes quite variable, the tumours get very big, and we were finding that we were missing taking animals because they were dying, and we sort of didn't have the hands on the ground to do it and that kind of thing. So most of the data that I'm going to show you, the mice were taken at 14 days. But this is data from 16 days, and we get significant loss of body weight, loss of fat mass, and of skeletal muscle mass. We find we can't use either DEXA or NMR for lean mass in this model because it picks up the tumours, and the tumours are quite large. So we really are relying on dissection of individual muscles to get a readout of lean mass or skeletal muscle mass. So as I mentioned, one of the main signalling molecules that tells the brain about hunger or about reduced energy availability is ghrelin. In case you're not super familiar with ghrelin, ghrelin must be acylated or active to bind to its receptor, the growth hormone secretagogue receptor. So it circulates in two forms. The inactive or the desacylated form is much more abundant than the acylated form, and it's acylated by this enzyme called ghrelin-OA acyltransferase, or GOAT. And so to really get a good readout of what's happening in your system with regards to ghrelin, you need to measure both total ghrelin and also active ghrelin, because just having total ghrelin won't tell you what ghrelin is doing in your system. You need to know how much of it is acylated. And then you can look at a ratio of those two to see if there is a sort of an issue with, say, acylation or the overall balance of the two forms of ghrelin. And so we took animals, so either PBS treated animals, so sham, or animals on day 4, day 10, and day 14. So just to take you back here, day 4 is no weight loss. Day 9, 10 we find is really this tipping point of the disease where the animals go from being like pretty normal to dropping off in their food intake and body weight, and then day 14 where we've got significant disease. And we find that ghrelin levels, if anything, are increasing, but they're certainly not decreasing in these animals. So the ghrelin is there to provide support for the reduced body weight, but it's not acting. This is the body weight data just that matches this cohort. So you can see that really on day 14, the animals have lost a significant amount of body weight. And interesting, I think, and this, I'm still thinking about this and what it might mean, but someone kind of mentioned it to me yesterday as I was putting this talk together, and I thought it was an interesting point and worth bringing up. This is a different cohort of animals, but we just measured blood glucose on day 10 and day 14 just to see what was happening there. And it was sort of unsurprising to us that the blood glucose was reduced, given that these animals weren't eating all that much and they were losing weight. But the really interesting thing about ghrelin is that the one thing that it seems to be quite critical for is maintaining blood glucose levels during starvation. In the early aughts, they did a bunch of sort of ghrelin knockout studies, and basically the thing that came out was that if you calorie restrict animals that don't have functional ghrelin signaling, they can't maintain blood glucose in the face of not having calories coming in, and they actually die of hypoglycemia. So I'm wondering if this kind of maintenance or slight increase in ghrelin is actually reflecting changes in blood glucose and really doesn't have anything much at all to do with appetite regulating circuits. So this is something I'm going to think about more, and I wonder if maybe the conversation around ghrelin has maybe been happening a little bit off target. So just to then test this functionally, we wanted to see, well, okay, they have normal circulating ghrelin, but can they respond to a ghrelin challenge? So this is data from some time ago now that I produced that's just, I mean, many people have done this, but it's just a dose response of ghrelin and looking at 2-hour food intake following a ghrelin injection. And you see a really nice increase in feeding when you inject them IP with ghrelin. And this, once you get up to 1 milligram per kilo, you can see that the amount of food they eat is analogous to sort of an overnight fast. They really, and these are satiated mice in the light phase, so they wouldn't normally be eating. So it's quite a potent driver of feeding. So we wanted to then have a look at these two critical time points at day 10, and we chose day 13 here, but at the tipping point of disease and then when the mice are noticeably unwell, can they still respond to a bolus of exogenous ghrelin? And so for this experiment, all mice received a ghrelin injection, but they were either sham treated or they were PDAC bearing. And for both day 10 and day 13, we get a significant reduction in the amount of food that these animals eat in response to ghrelin, indicating that that signal is just not getting through. It's not being translated to meaningful behavioural change that will drive feeding in these mice. So one of the questions we had is why? What's happening? What is the breakdown in communication here? And so a few years ago, we got another component to the ghrelin system. So LEAP2, or liver-enhanced antimicrobial peptide 2, is an endogenous inverse agonist to the GHSR. The thing to sort of note about the GHSR is that it has really high constitutive activity. So about 50% of its signaling activity is happening at baseline. And so for a long time, we had this system through ghrelin where we could modulate activity up and increase the activity of this already active receptor. But we didn't realise that there was also the other half of the equation where there is this endogenous inverse agonist that will reduce this basal activity. And so now we've really got an interesting balanced system where you can modulate this kind of constitutive activity up or down using these two circulating peptides. The interesting thing about LEAP2, we don't know a whole lot about it yet because it's been described so recently, but it is elevated in a number of inflammatory conditions, including rheumatoid arthritis. So it seems like it may well be relevant for cachexia. And so we went ahead and measured it on day 9, and this is pooled samples from across experiments, and we get, excuse me, we see elevated LEAP2 in these KPC mice. The better experiment to do is to use the one we're planning to do, I just didn't quite get to the ELISA before this talk, is to use the data from the ghrelin data that I showed you previously, use the same plasma and match up particularly acylated ghrelin with LEAP2 levels and look at the circulating molar ratios within individual animals, and then we can get a snapshot of where this balance is sitting for each mouse, and hopefully we'll be able to get a cleaner picture of what's happening then with regards to LEAP2 activity in this model. But it does look like LEAP2 may be elevated and not allowing ghrelin to act in the way that it should. So LEAP2, regardless of whether or not it's elevated circulating LEAP2, the main target of ghrelin in the brain are AGRP neurons. And so we wanted to have a look at what was happening at the level of the brain. Are AGRP neurons able to respond to a bolus of ghrelin by increasing their activity when mice have cancer? And so to go about this, we're using fibre photometry. Excuse me. And I'm not too sure how familiar this audience will be with fibre photometry, so I've got the full explanation here, and apologies if it's boring. But fibre photometry is a method for measuring activity, a population level of activity in neurons in the brain. So it's an optical technique that we can use to record from freely moving alive animals. And it uses a fluorescent peptide, GCAMP, that links a GFP to a calcium calmodulin binding system. So it's a modified calmodulin and myosin light chain kinase construct. And when there's no calcium around, it sits in this position where the GFP is not fluorescent. When you get calcium influx into the cell, so when a neuron becomes active and there's more and then calcium enters the cell, you get binding of calcium to calmodulin, and the calmodulin myosin complex comes together, and it causes a conformational change in this GFP that allows it to fluoresce. And so we can measure light that comes off this GFP as a proxy for neuronal activity. And it's a really powerful technique for looking at neurons that have similar properties. So we can target them using some sort of genetic technique, using a sort of, say, a Cree system or something like that, where you can actually say, okay, we want to look at this population of neurons that all express the same neuropeptide. So this is the approach that we're using to look at the AGRP neurons. And so stereotaxically injecting adeno-associated viruses that have Cree-dependent expression of GCAMP. And then we can use the sort of a photometry system to pick up the activity of these cells while the mice are doing tasks. So this is pilot data that I'd like to share with you today. So please, there's no statistics on this, and obviously take it at face value. But I just wanted to share the technique and how we're really, we're going to drill down on the specifics of how brain function changes across the progression of cachexia in this model. So this is the kind of timeline that we use for each experiment. The mice are put into the arena for photometry recording and attached to the photometry cable, and they're allowed to habituate to that arena for about 15 minutes. We then give them an IP injection of ghrelin. We record for 45 minutes in the absence of food, and then we give mice food and record for another 30 minutes. So we can then extract the traces of collected light and analyze it as change from baseline as a proxy for how active those neurons are. And I'd just like to give a shout out to Alex, a really sort of talented senior postdoc in the lab who's really been helping me with these experiments and who is quite the photometry guru. So just to take you through this graph, we've got baseline recording here, which is what everything has been zeroed to. We then have ghrelin injection at time 0, and we have baseline in the sort of dark purpley grey colour. Then we have mice at day 9 of the KPC model here in the light blue, and at day 14 in the pink. So again, pilot data, so grain of salt. But interestingly, it appears that the day 9, that the activity of AGRP neurons in response to ghrelin is reduced at day 9. So they're not picking up on that signal as well at that tipping point from kind of no obvious disease to obvious disease. And then possibly when they're becoming quite unwell, we're getting a stronger response to the ghrelin signal again. So it will be really interesting to have a look at whether or not there is dynamic change in these neurons across the time course as we are able to add more animals and really kind of tease apart these differences. But I hope that you can see here that this is a really powerful tool for being able to look at the specific neurobiology of what's happening in these feeding circuits during progression of cachexia and this model specifically. So we definitely know that these mice are not responding normally to ghrelin, the sort of main signal of negative energy balance. But the next question we were interested in asking is, are AGRP neurons able to respond and to do the things they normally do, so promote feeding and weight gain and energy conservation, if we can target them directly? So we went about doing this by using an approach called DREADS. Again, I'm not too sure how familiar you are. I'll do the quick and dirty explanation. And if it doesn't make sense to you, you'll just have to kind of take it on face value, that this is an approach that works. So DREADS stands for Designer Receptors Exclusively Activated by Designer Drugs. And the way we do this is to stereotaxically inject a Cre-dependent AAV, in this case into AGRP Cre mice. So we get recombination of this mutant receptor just into the AGRP neurons in the hypothalamus. The mutant receptor is this human mutated version of the human muscarinic receptor. It's a GQ coupled receptor, and it is activating, so it activates the neurons. We're able to activate it by injecting a drug called CNO, or one of the newer analogues of CNO now that are a little bit more selective. And by injecting this drug that has no ligand, no native ligand in mice, we're able to selectively activate just this population of cells that we have delivered this mutant receptor to. So it's a whole genetically modified system whereby we can use a a novel drug to activate a mutant receptor, and we are exclusively getting activity in the AGRP neurons, not in other cells in the brain. And we're doing it in the absence of changes to regular ghrelin signaling or other systems within the body. So why might we think that activating AGRP neurons would be a good approach in cancer cachexia? So this is data from the original Mike Crash's paper from 2011 now that described how DREAD activation in AGRP neurons works. And he's got a bunch of extra controls here, but really the comparison you're looking at is between red and all of the other animals. So when these mice have activated AGRP neurons, we get increases in food intake, we get reduction in oxygen consumption as a proxy for energy expenditure. When they're dosed repeatedly over a number of days, there's an elevation in body weight, which drops off when the mice are switched to saline injections. And we also get accumulation of fat mass and increased food intake over these longer term injections. So these are turning on these neurons is really pushing body weight in the direction that we would hope with regards to appetite in the context of cachexia. And we've got good data that the melanocortin system generally and the AGRP neurons are good targets for this. And a lot of that work has come from Dan Marks and his group. And this is quite old data now from 2001 where AGRP, so essentially it's the difference between these two lines here. I know this graph is not the easiest to read, that shows when you give just AGRP peptide into the brain, you get rescue of food intake in this tumour model. So there's quite a lot of sort of precedent for why targeting AGRP neurons would be helpful in cachexia. So we went ahead and generated these DREAD mice by injecting the AAVs, and then we ran them through our sort of standard 14-day, or actually some of these we took out a little longer, but our standard KPC protocol that I've explained to you already. And so this was work done by a wonderful master's student a few years ago now. But when we, sorry, just let me take you through the groups here. So we've got wild type PBS. So these are sham controls. We then have wild type animals with the KPC. So these are our cancer bearing animals. We then have animals with activated AGRP neurons, but without cancer. And the last group here is activated AGRP neurons with cancer. And so we start dosing on day 9, which is when we see the change in appetite. And we chose this time point to reflect a clinical situation where you might see more obvious signs of onset of cachexia. And when we do this, we see in mice that have activated AGRP neurons but no cancer, a significant elevation in body weight and in fat mass. But critically, in mice that do have cancer and have activated AGRP neurons, we also see this this actual gain in fat mass and gain in body weight and maintenance in fat mass. But this is possibly reflected simply by the fact that we've got more animals in this group. We didn't NMR every single animal that came through. So the fact, it may well be that this more modest effect here just reflects the fact that we've got fewer animals in this group. And if we look at, this is just this data expressed differently. And I'm not quite sure why I did that, sorry. Essentially, we get normalization of both body weight and fat mass when we turn on these AGRP neurons. And this was dosing CNO at quite a standard dose of 1 milligram per kilogram, and we did it twice a day, right around when the lights went on and when the lights went off. So the other obvious thing to look at is food intake. So turning on AGRP neurons increases food intake in both the sham-treated animals and in the PDAC-bearing mice. And interestingly, this effect seems to drop off over time. So it may be that initially when the animals aren't too sick, it's able to work quite well, but towards the end, cachexia wins, which is sort of the way it goes, I suppose. But some challenge, so measuring, I'm not sure how many people work in food intake, but measuring food intake in mice is quite tricky to do it well. And this model has a few additional concerns that make it a little bit harder again. One of those is that these PDAC-bearing mice shred their food, and they also tend to eat their bedding. So we're not, it's a little bit harder to measure what's actually going in. We've also found quite repeatedly that we see social transmission of behaviour. So the control animals seem to reduce their food intake in response to the worsening sickness of the cachectic animals. And that's controlling for that is one of those every way you can come up with to kind of control, you're introducing another confound as well, like housing them in another room, that kind of thing. So it's something that we just live with, but I don't think, in many of our studies, I don't think that the reduction, the difference between the control group and the cachectic group truly reflects the difference between a completely normal group and a cachectic group. I think that it's closer together than it maybe should be. And the other kind of consideration here is not specifically to do with food intake, but that AGRP neuronal activation and ghrelin as well has a primary anabolic effect. So in order to try and really get a good handle on food intake and look at whether or not this was simply due to increased feeding or whether or not it was due to promotion of anabolic processes, we've dived a little bit deeper into the feeding behaviour using these Biodac feeding cages. They're quite good at measuring spillage, and they've given us a bit of a tighter view on what's happening with these mice. Now, again, this data is a little bit preliminary, and I'm sorry I didn't get a chance to go in and tidy it up. It's sort of directly from an honours student who finished recently, but I think it will get the point across, and that is that in wild type animals, we see maintenance of circadian rhythm with feeding. So this is hourly average of food intake across the last 4 days, between day 10 and day 14 when the mice are sick, or not sick in the case of the sham-treated animals. And we see the sort of standard circadian pattern where the mice don't eat very much during the light and they eat quite a lot more during the dark phase. In the mice with activated AGRP neurons, we get feeding all through the light phase. So it seems like quite a lot of our effect on body weight is driven by just eating more food, which is not super surprising given what we know about AGRP neurons. So really just driving food intake when mice wouldn't normally eat is really helping these animals. So we also, though, wanted to look at the energy expenditure side of the equation. So we've had a look at bat temperature as a proxy for energy expenditure and locomotor activity. So the top panels are locomotor activity and the bottom panels is brown adipose tissue thermogenesis. Oh my God, yeah. And so this is the last 48 hours, so days, and actually it's the last 48 hours because this was variable time points. This was when we were leaving the animals go a little bit longer, and so we had to kind of work backwards from when they reached their ethical endpoint. So it's sort of been normalized to sort of decline rather than to time from injection. But what we see is a reduction in we see a reduction in locomotor activity in all groups compared to the sham PBS, sham wild type animals. Early morning is catching up with me. This is not surprising for the AGRP activation to do this both with and without cancer, in that we know that turning on AGRP neurons will promote locomotor activity in the absence of food because mice will look for food but when there is food, they just sit still and eat. So this lack of locomotor activity is likely just being driven by more time sitting at the hopper. We also know that elevated AGRP activity reduces brown adipose tissue thermogenesis, which we saw to some extent here, but not possibly not significantly. The main, hang on, am I getting this right? Yes, those difference between the sort of pinkish line, hang on, they're the cancer. Sorry, early. The point I want to make here is that the temperature of the wild type animals with cancer is the lowest. And even though we know that activating AGRP neurons has a tendency to reduce thermogenesis in normal animals, what we see here in the mice with PDAC is a normalization of thermogenesis. So this may simply be reflecting the fact that they have more food available to burn and their body weights are better. So even though they are essentially using more energy for thermogenesis, I think this reflects a normalization of physiology and is probably a good thing in this context. And as an aside, I'd just like to point out that this model, we really see a reduction in thermogenesis if we compare the dark blue line, the wild type PBS, with the red line, the wild type KPC animals. And this is in quite stark contrast to the, sorry, I'll just pop all this data up here, to the C26 model. So this is data that I generated for another group, well, 12 years ago now, when they approached us, and it was during my PhD when I was doing a lot of these brown fat recordings, but for obesity studies, they needed someone who could do them for their cachexia paper. And so the way they wanted to run the study was looking at PBS, which is the black line, compared to a weight yoked animal in blue. So these are mice that were calorie restricted so that their body weights were yoked to the cachectic mice, which are shown in red. And then they had the C26 ad libitum animals. And so in this model, we really saw an outrageous increase in bat thermogenesis, particularly when compared to mice that were calorie restricted, so they were losing the same amount of body weight. And you can see reflected in locomotor activity in the C26 model, we have this real elevation in locomotor activity in calorie restricted animals or the weight yoked animals here, where they're just running around trying to find food, which and we're really not seeing that reflected in the KPC. Well, we don't have a weight yoked group here. These mice all have ad libitum access to food. But my point, I guess, would be that different kinds of cachexia are different. And so I think there's quite a lot of variation in these models about the underlying physiology. And I'm not too sure how generalizable some of these sort of metabolic parameters are between models. And that's something that I'm really kind of interested in teasing out with the brain activation too. Like, what is the fundamental difference between, because we know that thermogenesis is largely controlled by the brain, what is the fundamental differences that are occurring between models that mean that the brain is responding differently in these two situations? So my little digression aside, we also looked at AGRP activation on skeletal muscle mass, and we see that in wild type animals, as I showed you before, we get a significant loss of skeletal muscle mass. But in animals with activated AGRP neurons, this is rescued. So mice that have cancer, activated AGRP neurons, their muscle weights are the same as the control animals. But one question we had is how much of this is due to food and how much of it is due to sort of direct neuronal activation. And it looks like an awful lot of it is due to just food going in, more substrate. So this, we air-fed animals with, so So these animals all have activated AGRP neurons, and here we have a group that is ad libitum fed, and we have a group that is pair fed to this group here, essentially. And when we do this, we see that activation of AGRP neurons is not effective at restoring muscle mass. We get a significant reduction in both the TA and the gastroc, indicating that we do need the increased food coming in to get this effect. And this is just, again, a slightly preliminary data, but I put it in here for this audience because I know you're all, this is everyone or many people's home ground here. But we also measured circulating levels of activin A and activin B in these mice, and we essentially saw no difference between. We saw a big difference when mice had cancer, but there was no difference when mice had activated AGRP neurons. But intriguingly, when we did the PCR, we're getting, I know this, it's not significant. The animals, the numbers are small here in this experiment. We're getting a suggestion that possibly the effect on muscle is disconnected from this very high circulating levels of activins. So this is something that we're going to expand on and have a look at whether or not there might be a direct effect of AGRP neurons on muscle, independent of these circulating ligands for the activin receptor. So that was my last piece of data. So in conclusion, I'd just like to say that the KPC model may be a state of ghrelin resistance, and that directly activating AGRP neurons can increase fat mass, skeletal muscle mass, and body weight. So if we hit those neurons hard enough, they can do their job, but they're not responding the way they normally would in a physiological situation. In addition, AGRP neuronal activation increases food intake in mice, and this is likely driven by elevated intake during the light cycle. And we're pretty sure that the anabolic effect of AGRP neurons on skeletal muscle is secondary to food intake, which is perhaps not as exciting as having a primary anabolic effect, but I think just having the substrate go in is really possibly more important than I realised before I started this work. And lastly, that activating AGRP neurons normalizes bat temperature and reduces locomotor activity and returns the animals to a more sort of homeostatic looking situation. So I'd just like to say a big thank you to my funders, as always, and particularly the people, the students that have worked on this project over the time, and Alex and Kelly, who have both really kind of supported me in getting various, and sorry, also Erica and Adam, who've supported me in really getting parts of this project off the ground. So thank you very much.
Thank you, Sarah. Terrific talk and very, very fascinating results.
Thank you.
Thank you. So there are a couple of questions in the Q&A. So the first one is if you checked for some functional test as the grip force, for example.
So we did sort of voluntary grip strength, the one where you sort of put the mouse on the little grid and pull on their tail. We didn't see any consistent changes. I don't know that I have an awful lot of faith in that data, just because it's difficult to do it repeatedly, and it was a student doing it too. So, I mean, that always adds a little bit, a layer of complexity to the data. But we have just gotten, through an equipment grant here, an organ bath that will allow us to actually test ex vivo muscle. So going forward, I hope to actually be able to test it that way in a more sort of controlled way.
Thank you. Then there are another couple of questions that are connected. So the first, did you evaluate survival? And if you see change in mortality rates in the mice with manipulated AGRP neurons?
So we don't see differences in tumour weights or survival generally that we can kind of observe just by cutting the tumours out and weighing them. With Erica, we did a time course to look at whether or not so using bioluminescence to image the tumours and look at tumour size across time. We saw no differences until the very end point when AGRP activation seemed to make the tumours bigger on the very last day, day 14. The one caveat of that was that it was in female mice, and most of the rest of the data that I showed is in male mice. And I suspect there's maybe some sex differences to the cachexia at least. I'm not too sure about the cancer progression, but I'd like to repeat that study in male mice just to have it match with the rest of the data set.
Okay, then there is another question. Have you measured LEAP2 levels at day 16, which is the kind of similar to the end stage of cachexia?
So the current, I haven't, no, is the short answer, but the current plan is to use the bloods that are matched to the ghrelin that I showed you, which would be day 4, day 10 and day 14, so not quite end stage. But I would love to hear, so I guess part of presenting to this audience always is to get feedback about what would be useful and what would be the best approach. So thank you for that feedback. I won't be able to do it in that cohort where I've got it matched to ghrelin, but when we do leave animals out a bit longer, I will go ahead and do LEAP2 in those bloods as well. Thank you.
So before going on with the other question, I have another one. So looking at your data overall, what is your feeling regarding the importance of food intake versus metabolic processes in the KPC model?
I think food intake's really important. I was really, I mean, we all want the exciting finding, right? And I was really hoping that we would get an anabolic effect or something that we could pull out of AGRP neurons independent of food intake. And it just doesn't look like that's the case. It really looks like it's the additional substrate going in that's driving this. And we're currently kind of failing to do an experiment, failing to do a very simple experiment. Just that's how these things go sometimes. But where we're just trying to match the timeline. So from day 10 to day 14, we give the mice access to Ensure and just look at what happens when we are able to, and they're voluntarily drinking it, they're drinking quite a lot of it. The issue we're having is that the control animals don't seem to be developing tumours as well as they should be. But they're getting fat just with extra Ensure coming in. So I'm hoping that we can kind of pull that apart a little bit, because I'm actually starting to suspect that really it's the food intake that's the important thing here, so much as other kind of metabolic processes.
Thank you. So another question is, did you measure GHSR transcript in the AGRP cells and or elsewhere?
We haven't, no. What I'm kind of hoping to do, so we have the GHSR pre, and there's sort of other projects ongoing in the bigger lab group that are looking at deleting the GHSR off the AGRP neuron specifically, and having a look at whether or not that changes function. So I feel like that would be a really nice additional piece of data here, is to actually just selectively ablate the GHSR off the neurons and have a look at how that changes.
Thank you. So another question is about MC4R antagonist that has received attention for cachexia treatment. So has your AGRP data influence your thinking on MC4R as a target in cancer cachexia?
Look, I think it's worth trying. And I don't work with patients, and there's probably people in this audience that have much better understanding and feel for this than I do. But one of the things that the people I have spoken to seem to report is that is sort of upsetting to them is that loss of ability to participate in meals and to enjoy food and to kind of even feel motivated to eat. And I think these are all things that we know the melanocortin-4 receptor, or the melanocortin pathway is good at, is sort of promoting feeding generally. And I feel like even regardless of anything else, getting food in and allowing people to kind of participate normally in meal times and in feeding events, I think would be really helpful. But I am very much a basic scientist. So I feel like the translational questions are a bit sort of iffy for me.
And do you have any hypothesis about the molecular mechanism who is leading to this absence of answer from this specific subpopulation of neurons to ghrelin? Do you have any kind of hypothesis or idea about the mechanism underlying your
I think the problem is I have many. I don't know what my favorite one is. I know, well, these neurons have can respond to IL-6 and IL-1 beta. So sort of elevating circulating levels of cytokines might be quite important. We're pretty sure it's not activin. So I did a series of experiments where looking at, because there was some early reports from a long time ago that indicated that activin can suppress feeding. And so did a series of experiments looking at that. Activin can suppress feeding, but it doesn't seem to suppress ghrelin-induced feeding. So however it's doing it independently of the AGRP neurons. I kind of feel like, in the words of a Spanish postdoc I had years ago, they get messed up somehow. I think there's probably, because they're so close to the blood-brain barrier, also privileged, I suppose, they're at that first line of getting all of these circulating factors that we know are kind of problematic for a range of tissues. And I kind of suspect there's inflammation, they're just not working properly. They're not able to kind of get it together.
But related to this, beyond the activins, did you measure the level of other inflammatory cytokines or GDF-15, for example?
Yeah, so we measured GDF-15. GDF-15 goes up when we compared sham to PBS. Sham to PDAC.
Yes.
So essentially it goes up in the KPC model.
Yeah.
When we looked at it in the mice with activated AGRP neurons, we actually saw that it seemed to go up with surgery. So I think, we never dug into this deeper, but essentially all the mice that were in those AGRP studies had higher GDF-15 than the mice that had never had any intervention. So my hypothesis there is that the GDF-15 goes up with the surgical burden, and then we start the cachexia sort of 2 to 3 weeks later after the dreads have expressed, and it just doesn't have time to kind of come back to baseline and normalize. And so it's just kind of chronically elevated over that period.
Okay. There are a couple of other questions. So since AGRP stimulation reduces the locomotor activity, would it make sense to test the animal exercise resistance in either non-tumor and tumor-bearing conditions?
It probably would, yeah. And they will, I guess, I'm just trying to think, they will move more if they don't have access to food, but I don't think that's not going to help. Not having access to food. So yeah, I guess I hadn't really thought about exercising them, but yeah, that's quite that's an excellent idea. Thank you.
Okay, so the last one, have you considered treating as well with chemotherapy to see the impact as a sort of combination therapy, and potentially have a food restricted control group closer to the tumor bearing intake?
Yeah, so definitely considered it, and it's in the grant, but yeah, just haven't got to it yet. But I do think it's obviously a really important consideration, because we know all these things impact as a unit, right? So yeah, absolutely. planning on trying to get those studies done and have a look at how that all works together.
Okay. And in the muscles, did you also look at other catabolic pathways, such as autophagy, for example, beyond atrogen and MERF1?
We haven't, no. We had a bit of a go at fibre size and have not been super successful at getting that data analysed, but we're still working on it. But yeah, no, Adam's been helping me and he's been really wonderful. But yeah, muscle's not my kind of home ground. So, but I appreciate that there is more to be looked at there.
Yeah.
We can at least do it with PCR, even if we can't sort of do it with other things.
Yeah, yeah. Okay, Sarah, the discussion was very lively, so I'm very happy for that. Thank you.
But not too lively.
Unusually lively, so it's a good sign. Thank you, Sarah, for accepting our invitation and for your talk. Thank you, everyone, and see you next month for the next seminar. Thank you, everyone.
Thank you very much, everyone.
Bye-bye.